Okay,
And
I'm
then
recording.
Dr. Narla, you should be able to share.
Take two. I look all right?
Yes. All right, everybody. Thank you guys for coming. I hope you all have been doing well in quarantine and the state of everything going on in the world. And I just hope that everybody is safe and going to get back to lab soon. And we're figuring out how to deal with this new normal. But I really want to thank Dr. Narla and Dr. Alan Peterson for both presenting today. So to kick this off, we'll start with Dr. Narla. Dr. Goutham Narla is an associate professor in cancer biology and chief of the Division of Genetic Medicine at the University of Michigan Medical School in Ann Arbor, Michigan. And today he's going to be discussing the role of PP2A in cancer treatment. And with that, I will let you take it away, Dr. Narla.
Great. Thank you again for organizing this. This is great. And while we can't do this in person, at least we get to share our work with our colleagues in the field. So today I'll be talking about turning on the brake for cancer treatment and lessons we've learned in my lab from protein phosphatase 2A. Oh, sorry. You think after. So I do have a few disclosures to make. I'm a co-founder of Reactivatable that's developing small molecule activators of PP2A for the treatment of a broad range of human diseases, including cancer, and have patents associated with that work, as well as a scientific advisor for Hera Biolabs. So as everyone here is very well aware, reversible phosphorylation of target proteins is absolutely critical in the regulation of a diverse set of cellular processes. And the hijacking of that in diseases such as cancer leads to pathogenic cell proliferation and aberrant cell survival. Our group is very interested in protein phosphatase 2A for a number of reasons that I'll outline in a minute. Just get the laser pointer here. What's really critical about this particular family of serine threonine phosphatases, and I want to emphasize that it's a family, is that there are about 20 different regulatory subunits, which give it incredible structural diversity. And that allows it to dephosphorylate many different substrates at the cell simultaneously. And each one of these really represents a member of this ever-growing family of serine threonine phosphatases. By having so many different cellular substrates, it can regulate many key processes involved in both normal physiology as well as in pathophysiological processes. And I'll focus on cancer today for our talk. Seminal work by a number of people here in the phosphatase field have defined PP2A as a tumor suppressor. Marine toxins, which are potent catalytic site inhibitors of this phosphatase, in fact, are tumor promoting. Bill Hahn and Bob Weinberg and many others show that DNA tumor viruses, many of them code for specific antigens that in fact inactivate PP2A and drive tumorigenesis in both cellular as well as in vivo model systems. The process of biogenesis has been worked out by David Pallas, Egon Ogrist, Dave Vershup, and many others in the field, and it's really a fascinating process whereby the heterodimeric protein of PP2A can load any one of these 20 different regulatory subunits. And in fact, the carboxymethylation in a unique reaction driven by LCMT of the terminal leucine neutralizes a charge which allows specific tumor suppressive regulatory subunits to bind. And this will become important later on as we talk about how this is subverted in cancer, as well as how some small molecules that have been developed by my group and others actually can activate this phosphatase family in a very specific manner. So really the first lesson that we focused on in the lab in terms of study is to just go through the TCGA data and ask ourselves, are there recurrent mutations in PP2A in the family? And if so, what members of the gene family are these recurrent mutations present? So a couple of interesting observations come out of that work. The first is that the most frequently mutated gene in this family is the scaffolding subunit. And there are 2 recurrent mutations in the scaffold subunit that are present in about 30% of a certain subtype of uterine cancer called high-grade serous endometrial cancer or uterine cancer. And these mutations actually exist in almost no other human cancers. So they show a tremendous amount of tissue specificity. Work done by Gibson et al. Had shown previously in sequencing efforts that these particular mutations that we'd identified, in fact, are truncal, meaning that they're present in both the localized tumor as well as in the metastatic foci in these patients. And they co-occur with almost complete penetrance with TP53 mutations. We went on to solve one of the first high-resolution structures of this mutant protein, and in this case, this was solved at about 3 angstrom average resolution with Wenqing Zhu, and you can see a marked conformational alteration in the mutant scaffold compared to the orange, which is your wild-type scaffold there. That mutant then is associated with decreased enzymatic activity. And what's interesting, what we hypothesized is, given the nature of the change in the scaffolding subunit imposed by this mutation, we predicted that it might disrupt catalytic subunit binding. In order to test that hypothesis, we went on to make isogenic cell lines overexpressing V5 tag versions of this mutant. And what we see here is that there's a 80 to 90% reduction in the ability of the catalytic subunit to bind this mutant A alpha scaffold, and that's probably driven by this conformational alteration imposed by that mutation. So the next question, of course, is this mutation pathogenic? We would assume that it would be, but in order to test that hypothesis, in collaboration with Annalisa DeFeo, who has a large collection of endometrial cancer PDXs and cell lines, we identified several patients who harbored this mutation and were able to derive both patient-derived xenografts and cell lines. One of these particular patient cell lines that we derived and have characterized, we introduced the wild-type form of the scaffold, and you can see marked tumor growth inhibition both in a subcutaneous model as well as suppression of metastasis in an orthotopic intrauterine model by reconstitution of the wild-type scaffold in this context. That in aggregate suggested to us that this mutation most likely is pathogenic. Interestingly, the TP53 conditional knockout in the endometrium develops moderately differentiated serous cancer, but doesn't develop high-grade metastatic disease. So we've actually generated a P179R mutant transgenic mouse now, knocked in that allele, and we're thinking of crossing that to the TP53 mouse to create potentially a unique gem model to recapitulate the entire spectrum of this deadly disease. Next, what we looked at is another common mutation. In this case, this occurs in 0.6% of all human cancers, but unlike the other mutation that I showed you, it's kind of widely distributed across human cancers, with colon, lung, breast, and others having evidence of this mutation, R183W or Q, again, in the PP2A Aa scaffold. So an incredibly talented postdoctoral fellow in my lab, Caitlin O'Connor, went on to show that this mutation loses its growth suppressive and tumor suppressive activity. And unlike the other mutation that we talked about together, which disrupts the scaffolding subunit binding, this particular mutation really is associated with a loss of an ability to be carboxymethylated. Remember again that carboxymethylation allowed for the tumor suppressive regulatory subunits to load. And in this case, because you have decreased carboxymethylation, this was done by both mass spec, as well as using a proprietary antibody developed by Egon Obrist, the 7C10 antibody that's methyl specific. So you have decreased methylation, probably because of loss of LCMT1 binding, and that drives the loss of B56 alpha binding, which is one of the major tumor suppressive regulatory subunits that's been shown to be methylation dependent. This particular mutation is also inactivating in that it loses its enzymatic activity. And if we map those particular mutations back to the solved structures, one from our group and others from Wenqing Zhu and Yigan Shi, all of these mutations really sit at that interface of where the regulatory subunits come in and bind. And in many ways, phenocopy what the DNA tumor virus SV40 does. So SV40 displaces regulatory subunits in a mechanism that's very similar to these recurrent tumor-derived mutations, which I think from an evolutionary perspective is quite interesting. So if biogenesis is disrupted as part of the process of cancer development and progression, the question is, can we develop small molecule modulators to really promote heterotrimerization, reactivate this tumor suppressor, and leverage that for the treatment of a broad range of cancers? So one thing I want everyone to understand is, while I did talk about genetic mechanisms of inactivation of PP2A, these are in general across human cancer fairly rare. In fact, the majority of human cancers demonstrate loss of carboxymethylation or overexpression of some of the endogenous inhibitors of PP2A. And so they inactivate it in non-genetic mechanisms, which allows for unique pharmacological approaches to reactivate it therapeutically. So we know that kinase inhibition has been very successful in a number of different cancers to drive durable activity. Having said that, we would hypothesize that it would be equally efficacious to activate phosphatases, or the natural brakes on cancer cell growth, in order to therapeutically target that disease. In order to do that, we leveraged data from phenotypic screens done by our group and others that really was based upon a repurposing effort of a tricyclic series of neuroleptics that we had screened that were regulators of the FOXO Krupel-like transcriptional network. I'm not going to go into this in great depth because this has been published, but effectively what we and Tom Look's group and others were able to define was that these tricyclic neuroleptics, in fact, were able to bind and activate PP2A. Now, they're associated with profound GPCR pharmacology, so we went on over years to derivatize these to produce about 600 different analogs of these neuroleptics, in which we decoupled their GPCR pharmacology from their PP2A activating activities. These particular molecules drive pretty significant tumor growth inhibition in KRAS mutant xenograft models, as well as PDX models. This is work done by a staff scientist in our group, J.S. Sangokar. And many groups, including Rosalie Sears, Brittany, and others, have gone on to use these activators across a broad range of different tumor models with activity being noted, both as single agent, I should say, as well as in combination. By no means am I suggesting that we're the only group that has developed methods to activate PP2A. In fact, there are...
We'll just
recording.
Sandwich them together.
Yes.
Okay.
I'm going to exit the full screen for a minute. I'm just thinking. And do I have control of these slides?
Caroline.
Caroline, I can't advance them. Is that on my end?
Sorry, I was muted and I didn't know it. I'm adding you back. Just 1 second.
No worries. Or you can advance them for me. Might be safer.
I don't know if I can do that.
All right, no worries. Let's try it again.
Okay, you should be able to share.
Yeah.
Okay.
I'll jump to the slide I was on.
Okay.
Sorry, everyone. Can everyone see that?
Yeah, we're good.
Okay. So as I said before, by no means are we the only group targeting PP2A. There's a number of different approaches using either direct activators of this particular phosphatase or methodologies that indirectly inhibit the inhibitors of it to reactivate the phosphatase. And all of those have shown efficacy across broad ranges of cancer as well as non-oncology indications. So we really wanted to better understand the mechanism by which these small molecules were activating PP2A and whether there's any specificity to this approach. So the first clue that suggested to us that these molecules might be promoting heterotrimerization was work done in which we overexpressed the small t antigen, which is, again, I told you before, disrupts regulatory subunit binding. And in that particular context, the cells in vivo became resistant to these PP2A modulators. Dan Leonard, in a really talented MSTP student in our lab, and Wei Wong and Derek Taylor's lab, and mine went on to show in vivo that if you treat with these PP2A modulators and then you 2 hours post single dose pull down with V5 and look at the interactome in an unbiased manner, the only regulatory subunit that was increased in binding when treated with these PP2A modulators was B56 alpha. And that was interesting to us. And the question is why and how is that possible? So again, in collaboration with Wenqing Zhu and Derek Taylor, we're able to obtain recombinant A alpha, B56 alpha, and C alpha. We incubated them in vitro or in solution. And the first thing Derek noticed by SEC is that this heterotrimer falls apart quite easily. But when you add the molecule, the majority of it now becomes heterotrimeric. And that property allowed us to solve, using cryo-EM, the first structure of B56 alpha. So this is an average resolution of 3.6 angstrom across the 150 kilodalton structure. This structure has never been defined before. And what we're able to do at that resolution is actually identify exactly where the small molecule is bound. And it's bound right at the interface of where the scaffold subunit, the regulatory subunit B56 alpha, and the catalytic subunit come together. So it almost serves as a molecular glue that brings them together to promote heterotrimerization. Now, in order to test the validity of that particular model and where we position the molecule in the structure, we're able to, with George Trainer, a very talented medicinal chemist, and Nilesh Zawari, develop analogs that we would predict based upon the structure would not be able to bind. And these molecules are very structurally similar, as you can see, to DT61, but they either have methyl groups or benzene groups that would block key sites of interaction. Those molecules are completely inactive in cell culture and do not affect the affinity of B56 alpha to its target, further validating the structural data that we're able to derive. So the next question really is then how, if it's binding in a pocket and the B prime regulatory subunits are quite well conserved, where does specificity then, if any, come from? Well, it turns out it's absolutely fascinating that B56 gamma and delta, for example, have a lysine instead of a threonine at this position. This lysine allows for the formation of a salt bridge that actually occludes the site in which the drug accesses and binds. So what we're able to then do is, one, is we're able to show using SPR that these small molecules prolong the K-off rate, so they keep it together longer, prevent it from falling apart, but that's specific to B56 alpha. They have no effects on gamma delta. Then what we did is we made B56 alpha look like gamma by making that 1 point mutation, and now we lose the ability by SPR to see these differences. So that really suggests to us that this pocket is unique to certain B prime regulatory subunits, which is why we're driving specific heterotrimerization, or what I should more accurately probably say biased heterotrimerization in both cellular as well as in vivo systems. And I think that's important. The biology that we're driving could just as well be a product of increasing B56 alpha directed, say, MYC, dephosphorylation and degradation, work that Brittany and Rosie have really defined and the field around, or they could be because we're biasing it away from B55 alpha, and that's something we're very interested in further exploring. So kind of in the last minutes, I'll just finish with the following, is how do we measure this whole process is actually occurring, this biased heterotrimerization. Here again, working with Egon Ogris, we're able to develop this 7C10 methyl-specific antibody that we published recently in Cell Reports. And what that antibody allowed us to do in vivo is to show that with single-dose PP2A activator therapy, we're able to drive increased carboxymethylation because of the bias towards the methyl-dependent subunits. And that resulted in the degradation of MYC, one of the best characterized substrates of B56 alpha. Now, if we go full turn now, let's go back to that mutation I talked to you about before, R183W. If you remember, that mutant could not bind B56 alpha because of a loss of carboxymethylation. So interestingly, and I think a validation of the proof or a validation of the MOA or the mechanism of action is that mutation is actually completely resistant to PP2A activator therapy in vivo because we can't drive an increase in carboxymethylation that we can with other mutations or with the wild type scaffold. So in these contexts now with B56 alpha and MYC, we're driving the dephosphorylation and degradation of MYC. And so in collaboration with Rosalie Sears' group, we were able to show in triple negative breast cancer syngeneic models significant single agent activity with these PP2A activators as shown here. Oops. Sorry. So just lastly, that activity of those molecules is dependent to some extent on the ability to dephosphorylate and degrade MYC, because if we make phosphomimetics of MYC in this context and make isogenic lines, you can see that a lot of the activity of our PP2A activators is now lost. By no means is it all activity, so there's going to be other targets, but at least a significant, more than 50% of that is lost in these isogenic contexts. And so lastly, we can begin to say we have a PP2A modulator. How would it look in combination with other approved drugs? So with Yuka Westermark's group, we did a kinome screen. And so in this case, we screened for about 240 different kinase inhibitors and looked for synergies. And the top predicted synergy was the combination of a PP2A activator with a MEK1/2 inhibitor. And so we tested that particular combination in a preclinical series of experiments as shown here. This is just one example of that data. And we can see that the combination of a PP2A activator with a MEK1/2 inhibitor in purple here has more activity than either agent alone. And that work is published. So I would just like to end with the fact that really tumor-derived mutations in DNA tumor viruses have taught us a lot about how biogenesis is at some level subverted or hijacked to dysregulate this phosphatase and drive tumorigenesis. And we can leverage some of those processes using small molecule chemical biology tools to reactivate this phosphatase in a directed manner against oncogenic substrates like MYC. And the hope is that these tools ultimately would be translated to the clinic for patients for cancer patients. And so I've tried through the talk to acknowledge all the people in the lab. I'd especially like to thank Derek Taylor, a brilliant structural biologist and colleague, as well as Egon Ogris and Rosalie Sears in her lab. So thank you, and sorry for the technical problems.
Thank you, Dr. Narla. And we'll give you a virtual round of applause. That's the one thing that's always missing from these Zoom meetings. But we can go ahead and go with questions, and you can either unmute yourself, or if you want to send it in the chat, I can read it aloud. So.
Yeah, so I have a question. For the different A subunit mutations, do you see kind of heterogeneity in those mutations? Like, are there certain cell populations that have adopted that mutation versus others, or is it pretty much the tumor is all mutant?
Yeah, it's a good question, Brittany, and we haven't looked at that in great depth, I would tell you, by doing single cell sequencing or really trying to define subpopulations with those tumor associated mutations. I would tell you the truncality of the P179R mutation would suggest that it's a dominant clone in that cancer, and is selected for based upon its biology, but I don't have direct experimental evidence at a sequencing level to suggest that.
I have a question. Hey, Goutham, this is Haida Ford from Colorado. So I thought your data with your activator and B56 alpha was really interesting, and I'm sure you're aware of data that, again, you talked about this a little bit at the beginning, about how when it partners with B55 alpha, it can sometimes be tumor promotional. So do you think it's going to be as simple as if you have different regulatory subunits. It may be promotional versus inhibitory, and that you could similarly then think about developing inhibitors that break up the interaction with B55 alpha?
Yeah, it's a great question, and I mean your work has helped define some of that between B55 alpha and B56 alpha. It is quite possible that some of the aggregate effect on tumors here, Haida, is driven by the fact that we're inhibiting or relatively inhibiting B55 alpha. If you look at that interactome, it actually is there's less B55 and alpha in complex with our small molecules and more B56 alpha. Mm-hmm. Having said that, you could imagine trying to develop more selective B55 alpha inhibitors. I'm sure you're aware that B55 alpha loss has been shown to be associated with PARP inhibitor sensitivity, ATR1, CHEK1. So I think those are very interesting opportunities to think about exploring. And so at this point, I'm not sure which biology is dominating, whether it's the B56 alpha promotion or the B55 alpha loss.
Right.
And maybe you want both, and maybe it's actually advantageous to do both simultaneously.
Right. Thanks.
Yeah, thanks.
And then we had one question in our chat that's just asking if there are PP2A, I guess, semi or full deletions seen in any types of tumors.
That's a good question. So actually the homozygous loss of A alpha is lethal, embryonic lethal, and also it is almost never seen in human cancer. And Bill Hahn showed in Project Achilles that it's a common essential gene. It just isn't tolerated by CRISPR. We actually show that one of the only ways in which cells seem to be able to adapt to the complete loss of A alpha is by marked upregulation of the A beta scaffold, actually. And so that's a mechanism for compensation, but actually in sequencing efforts, there's almost never homozygous loss of A alpha occurring. Deletion of other regulatory subunits is seen, but in general, they tend to be heterozygous as opposed to homozygous loss in human cancer.
All righty. If nobody else has any other questions, thank you again, Dr. Narla. And then Jesse, if you want to stop recording and then